present in those sample. First of all, we have to do sampling. Yeah, not only in microorganism analysis, but also from the other analysis like chemical analysis, biological analysis, physical analysis. You need to do the sampling because you cannot analyze everything. Yeah. In food industry, you have many batch of your product, many batch of your raw material. Yeah, with a big amount. You cannot analyze a ton of your material or a ton of your final product. You just take sample. That sample should be representative for the whole batch. Yeah. Uh, you either analyze microorganism property in raw material. Yeah. In the food industry, when you produce any product, you need some raw material. Yeah. Like in the noodle company, the main raw material is flour. Yeah, with flour, something like that. And you have to analyze if there is any microorganism present in flour. Yeah, in flour or not, or the product of a candy. Yeah, you need sugar to make candy, so you have to analyze microorganism property in sugar. That's a microorganism analysis for material. How about during your production? You also do analysis, microorganism analysis for semi product or for final product. Yeah, each product, yeah, each sample, it is stored in different place. Yeah, like for raw material, you will buy raw material from the other company. Yeah, you buy sugar from the other company. You buy flour. You buy wheat grain. Yeah, from the other company, and you receive those material and store in a warehouse. Yeah, in a warehouse. So before your production, it should be stored in a warehouse. So QC staff will go to warehouse and take sample from the batch. Yeah. Or if you want to analyze semi product, the QC staff can go to the production line, go to the factory. Yeah. And your semi product, it is on the line. Yeah, on the convey line. You just take sample. Yeah, take sample if it is representative for that shift. Yeah, so in one factory normally there are 3 shifts. Yeah, morning shift, afternoon shift, and evening shift. So for morning shift, you take sample that representative for morning shift. Yeah, and representative for that line. Yeah, you have different lines of production. Okay, and if you want to take sample from final product, yeah, either you take from the line or you take from the warehouse. Yeah, your final product, it can be stored in the warehouse before transporting to supermarket or to the market for launching. Yeah, so sampling action is very important. Yeah, if you have a good sampling, that means you have a good result of analysis. Yeah, if you have not good sampling. So you analyze, okay, there is no microorganism in the sample, but it doesn't mean that there is no microorganism in that batch. So that's why you should have a good representative sample. Okay, on the screen you can see that there are two objectives of sampling. The primary objective of sampling that we need to collect food samples that are representative. Remember this term, representative. And then to ensure that changes in composition do not take place between collection and analysis. Yeah. When we take sample, it should be representative and it should be no change, yeah, between collection and analysis. Yeah, if you collect your sample and there is some change and then you analyze, so that analyze, the result of that analyze is on the change the change sample already. Yeah, it's not, it not indicate the result for the batch. Okay, so we should ensure that there is no change between collection and analysis. Yeah. The sampling that we need to write some information related to sample. Yeah, like season, geography, cultivar, husbandry. Yeah, we write and we document those information. Because with that information, you can analyze the result. Yeah, like if you do your thesis research, yeah, in the last year, yeah, in the last year when you do your thesis research, and you have your topic to do research, and you have sample to analyze, yeah. If you don't have any information related to your sample, if you do research, for example, you do research on uh, um, on the dragon fruit. Yeah, you do research on dragon fruit, and you don't provide any information related to dragon fruit. When you defend your research, the committee can ask you, yeah, why the result like that, yeah. So if you have information, you can base on that information. For example, cause dragon fruit that you bought is from the central uh, in the central of Vietnam, yeah, or in the winter season, yeah. So that's why your result different from the other result, yeah. So based on those information, you can have a good analysis, yeah. It's also the same for microorganism analysis. If you know your sample information, like you want to analyze milk, so you know information that milk is from where, yeah, you know the geography, yeah, some information related to your sample, yeah. So remember two objective you need to understand of sampling. I guess that you already learned statistic and you know the population, yeah, the population, yes. Yeah. Okay, so population with a big amount, yeah, like I mentioned earlier, a batch, for example, a batch. So now you want to analyze this population, Yeah, you cannot analyze the whole population. So you have to take sample. Yeah, like in statistics, yeah, you only analyze for sample only. You don't analyze for the whole. Uh, you don't analyze the whole population. But for sample, when you analyze sample, you can uh, have a conclusion for the whole population because that sample representative for the whole population. Yeah, this is the sample that we take from population. Yeah, and remember this sample representative for the whole population. Okay, so in industry, how we do a sampling? Yeah, um, we have two different kinds of. I hear the sound from. Okay. Yes. Your your sample, yeah, can be in bulk like this, yeah, or it is already packaged, yeah. That means like in some company like your sample, your rice, yeah, maybe they store rice in bulk, not in package, yeah, in bulk is uh, something like this, yeah, it's store like this, or sugar, yeah, it's store like this, yeah, it's different from the package uh, like this. So for the one in bulk, how you take sample, yeah, yeah, we can assume this is one lot or one batch, yeah, one batch of your sample. Now you can take sample, yeah, one in the left, one in the middle, and one in the right. So they are representative for the whole batch, for the whole lot. Yeah, the same for the package sample. Yeah, so this is big package. So inside there is some packaging. Yeah, you get one on the left, one in the middle, and one on the right. Yeah, and then you mix up them together, and you will get composite sample or aggregate sample here. Yeah, uh, this is for bulk, for the package. Yeah, the package here also contains smaller, yeah, smaller. So you just take random representative package inside here, get one, inside here get one, inside here get one, yeah, and even yeah smaller, yeah, and you will get, yeah, combine them together, and then you take from this, yeah, from composite sample you will take representative sample from this, and we call this is the secondary sample. Yeah, the primary sample is the sample that you take from the batch, from the lot. Yeah, this is primary sample. Yeah, and this is secondary sample. Secondary sample, and from the secondary sample, you won't do analysis for the whole sample here. You just take some to do in the laboratory. Like when you go to your laboratory, so TA, yeah, an instructor already prepared some sample for you, and each group will take sample, yeah, from the prepared one. Yeah. For example, we prepare a bottle of milk for you to do analysis. So each group will test sample from that bottle of milk. You want analyze for the whole bottle of milk. So the one that you take from bottle of milk, we call laboratory sample. Yeah, laboratory sample. Yeah, yeah. In some company, even they don't check the secondary sample. Yeah, they can check from the primary sample for analysis. Yeah, for analysis. And we have laboratory sample. Yeah, and we have a test sample. Yeah. Yeah. Each group will get sample from the big bottle of milk, and you will get a smaller bottle of milk for your group. Yeah, for your group, and then you will use micropipette to test sample for analysis. Yeah, so that amount of sample we call test sample. Yeah, test sample. And remember, in any analysis, we always convert sample into solution if that sample is not in liquid form. Yeah, if it is in solid form or semi-solid form, most of the time you make it into, you convert into a solution. Yeah, like if you want to measure protein content, yeah, of a meat, yeah, you have to make the meat sample into the solution, yeah, with some solvent, yeah, and then you will use a Kindan, yeah, the popular method Kindan system to measure protein content in meat. Yeah, we never use the Kindan with the bill, the meat particle only. Yeah, the meat should be inside some solution, and from those solutions you can get the result. So here it's the same for microorganism. Yeah, if you have to analyze microorganism in meat, yeah, so you will make meat to become solution. Yeah, you cannot use the fresh meat and you incubate the fresh meat together with medium, yeah, culture medium, and then you get the result. No, you have to make it into a solution. Yeah, we call alicate. Yeah, alicate. Yeah, for measurement. Okay, so this is demonstrate how we get representative sample from the big population like this. We get, yeah, some on the left, some in the middle, and some on the right. So they are representative. Yeah, it's different from that you just take some on the left and you do analysis. So maybe on the left there are no microorganism, but on the right or in the middle there is microorganism. So your result can be biased. Yeah, and you cannot conclude, okay, this batch contain no microorganism. Yeah, there are some terminology that you need to understand, need to remember. Yeah, in the previous food microorganism lab work, at the end, I didn't ask students to do the report, but I asked them to take an oral examination, and in those examinations, I will ask question relates to those. Yeah, and they have to learn by heart. They have to remember what is laboratory sample, what is test portion, what is initial suspension. Okay, so you are fortunate that you don't need to learn by heart here. But in your report, the requirement can be high, can be more than the one who take oral examination. Okay, so what is laboratory sample? I already saw laboratory sample here. Yeah, the sample that we take to the laboratory for analysis. Yeah, sample prepared for sending to laboratory and intended for inspection, yeah, or testing or okay. And what is test portion? Test portion is the measure either volume or mass. Yeah, we have the laboratory sample, and we get for sample 1 milliliter, yeah, of milk from the laboratory sample to measure, yeah, to analyze. So 1 milliliter here is the test portion. Or we have a laboratory sample is 100 gram of meat. Yeah, yeah. You have meat. You cut a piece of meat for your group to analyze, and your group will prepare the test sample. So they can weigh 20 grams of meat for analysis, and they take 20 grams of meat, and they make meat to become meat solution, or meat juice, or meat juice, and do the analysis on meat juice. Okay, so that portion is the measured either volume or mass representative sample taken from laboratory sample for use in the preparation of the initial suspension. Yeah. Initial suspension, we will talk about it many times during our analysis. So remember, what is it? Initial suspension, it's simple. It's the first dilution. Yeah, when you have the 20 gram meat, yeah, you put 20 gram meat into a flask, yeah, and you add diluent here, yeah, you add 225 diluent, yeah, you mix the meat together with the diluent, yeah, diluent here we can use buffer beton water. You mix 25 gram of meat together with 225 milliliters of buffer beton water, and you mix them together. Yeah, for the juice of meat can be released, and this mixture we call initial suspension. This is the first dilution, yeah, or primary dilution suspension, yeah. Initial suspension is the primary dilution, suspension, solution, or emulsion. Yeah, we can call anything. Yeah, dilution, suspension, or dilution solution. Yeah, obtained. Yeah, this dilution, this solution can be obtained after you get a weight or a measured quantity of product. Yeah, as I mentioned that 25 gram of meat, yeah, under examination, yeah, and has been mixed with normally 9-fold quantity of diluent, yeah. As I mentioned that I add 225 milliliters of buffer beton water into a flask, and in the flask there is 25 grams of meat. So remember this rule ninefold. Yeah, ninefold. We can explain what is ninefold here. Dilution. And remember, dilution. Remember the rule. You always follow the rule. One, ninefold.
Nhìn hũ bánh thấy ớn.
Ninefold. That means nine times higher than the volume of the amount of sample. I can give you an example. Normally, you can see that we call 110. So, how can you 110? 110 is the first dilution. Yeah, we make the initial suspension exactly is 110. Yeah, the dilution factor here is the 10 to the minus 1. For dilution here. You have your sample. Here. So if you want to dilute at least 110, you have the tester here. You take 1 milliliter of your sample in Okay, so until now, do you have any question? Yeah, if you don't understand what is initial suspension, what is nine-fold rule, you can ask me. Any questions? Okay, no question. Can I ask you? Uh, the first laboratory, you already know some media that we can prepare and know some diluent. So can you tell me, uh, can you give me one name of a diluent? Who can answer this?
Uh, some diluent we have not that is buffer beton water and beton water. There are two
Yeah, very
diluent
good.
that
Yeah.
we use.
Okay, yeah, very good fact. Okay, fact one plus score for him. Yeah. And we need diluent to do dilution. Yeah, diluent to do dilution. Okay, so let's move to the next uh, technique that we when we do the sampling. Remember, in the lab, yeah, before lab, we test sample, for a primary sample and laboratory sample. Yeah, but inside laboratory we will take test portion. Yeah, we will take test portion here, and test portion should follow septic sampling. Remember, yeah, and this is a technique used to prevent contamination by sampling by the sampling method. Yeah, aseptic sampling involves the use of sterile sampling implements and container. You can see the photo. Yeah, the woman, she used a cloth. Yeah, to clean. Yeah, of course there is detergent or there is alcohol to clean the bench here. Yeah, and there is alcohol burner here, and she had a knife. Yeah, to cut. Yeah, to take test portion. So she sterilize the knife with alcohol burner, and then she cut. Yeah, during her action, it should be close to the alcohol burner. Yeah, it's on. Remember, and she cut it, and yeah, she gets some busy, and then she can weigh. Yeah, she can weigh. Yeah, this is the balance. The balance also close to alcohol burner. Yeah, and she will weigh to get test portion. Yeah, test portion here. Yeah, this is solid sample, so she can weigh 25 gram of solid sample. Okay. Yeah, you can see Miss Ying use forceps and scissors. They are in a plastic bag because they are already sterilized. Yeah, autoclave. So that's why she put scissor and forceps in a plastic bag. Yeah, so everything should be accepted. Yeah, remember. The blender already sterilized as well. Yeah, everything should be sterilized. Yeah, we need aseptic sampling.
We have here 225 milliliter. And I will then for about 10 seconds and see if it's homogenized or not. Okay, continue with another 10 seconds. Well mixed and blended. Now I will transfer it into a sterilized oven layer flask. Okay. After that, we have to cover it with a piece of for some holes. So what is here? Okay. So this is my 10 to the power minus 1 resolution. And I can take this back to my working area and do the serial dilution.
Yeah, so what do we call for these solutions? Who can answer? What do we call this solution?
Is it an initial suspension?
Yes, that's right. Whose answers?
My name is Amber.
Yeah, Andre, 1+ call for her. Yeah, initial suspension here. Yeah, and if you want to make the further dilution like 10 to the power minus 2, you just take 1 milliliter from this, yeah, add to 9 milliliter of 9 milliliter of diluent buffer, yeah, Becton water, yes. So you can get 10 to the power minus 2. Okay, so who can answer why do we need to do aseptic sampling? If we yes.
Also, because we are studying about the microorganism on our sample, in this case, it's great. So if we do our experiment in the not clean environment, our sample will be contaminated with other strain of microorganism, which lead to the full result in our observation of the final result.
Yeah, thank you. It's very good. Who answers?
Is we
Okay. Huy, 1+ call for Huy. Yeah. correctly. Yeah, we need to find out the target microorganism in our sample only. Yeah, in our sample, we don't need a microorganism from the environment. Yeah, we just need to analyze if our sample contain any um, microorganism or not. Yeah, so that's why we need everything should be accepted. Yeah, accepted. Okay, so there are some beside sampling, accepted sampling. There are some other things that we need to sterilize. Yeah, like Miss Yuen, when she did experiment on the equipment or instrument or the container that they are on sterilized. Yeah, so inadequate precautions should be taken to prevent microbial contamination of sample from external sources. Yeah, equipment and material like scissor, knife, scalpel, can opener. Yeah, spoon, forceps. Yeah, we need to sterilize them all. Yeah, and there are some techniques to do sterilization. Yeah, first, you can use dry heating. Yeah, like you use alcohol burner like this. Yeah, you just flame. Yeah, you just flame. You can flame the equipment. Yeah, equipment on the alcohol burner. Yeah, or you flame. This is the media container. Yeah, the media. So you flame. Yeah, you use dry heating. Yeah, or you use moist heating. Yeah, moist heating that you can use autoclave. Yeah, autoclave. Uh, Last week, I showed the video how use autoclave. Yeah, Miss Yuen demonstrated how to use autoclave, and you add some water to make steam. Yeah, so that's why we call moist heating. Yeah, to sterilize. We can use a pre-sterilized plastic tool. Yeah, we can buy this. Yeah, we can buy this. The manufacturer already sterilized and they put it in a sterilized bag. Or we can use ethylene oxide. Yes, yeah, ethylene oxide. Yeah, you know this is for sterilization. But and do you hear this compound familiar with you? Do you heard anything about this? Have you heard recently on news that this compound appear on news?
It's contaminated in the instant noodle, right? And
Yes, yes. Who is answering? Huy?
Huy.
Okay, Huy. Yeah, Huy 1+ score. Yeah, in AC Cook. Yes, AC Cook. Yeah, when they export the noodle to Ireland, and they found this compound in noodle. It's not good. This is only used for sterilization only. Yeah. Okay. Another method that you can use to sterilize radiation. Yeah. Ultraviolet radiation or filtration. Yeah, filtration with a small size of hole. Yeah, you can separate some microorganism. Okay, as we have two kinds of sample: solid sample and liquid sample. Yeah, solid sample we call heterogeneous sample. Liquid sample we call homogeneous sample. Yeah. For liquid sample, it's very simple. Yeah. Before you take test portion, you need to check. Yeah. Okay. So there is a question here. Why do we need to check for liquid sample or homogeneous for solid sample?
We can get more the distribution for the analysis.
Yes, very good. Who is answering?
My name is Huanan.
Quinnan?
Yes.
Yeah, Quinnan. Yes, of course. If you test sample, yeah, the test portion like we put the micropipette inside this, and we get on the top, yeah, for sample. So maybe on the top there is no microorganism, but there is microorganism at the bottom for sample. So we need to make a good distribution of microorganisms so that we can analyze. Yes. And there is a rule here for liquid sample that we check 25 times. Yeah. This is the demonstration how we take liquid sample.
Okay, so now I will show you how to do the sampling of the liquid sample. So, and I will also do the direction from 10 to the power minus 1 to 10 to the power minus 3, which copper metal water may be greater than the limit. Okay, so here I have a gold sample. Testing with 5 ml of copper metal water already labeled. The room number, the sample, and the filter. And here a vortex mixer. This one is a sterilized tea. So all the things here should be done in a sterile environment. So first I will start with this international trends. It was sprayed alcohol all over the piece here. So when it's dry, let it dry by light. Then you will lie on alcohol for a while. What we the flame to make sure that the kind of alcohol do not contain flavors. Okay, so first for the sampling technique, so this one is low. First I will rotate the low magnifying glass. Rotate it clockwise and anti-clockwise. Rotate several times. When you finish, so first you will take a tip, and I will use the micropipette 1 ml because I just need 1 ml from transfer 1 ml of milk, the alphabet water. So simply take out one tip, close it. Use your window finger, hold the cap of the blue bottle, and use your left hand to rotate the bottle. Open it. Then inflate the neck of the bottle twice. Okay. Now you will take 1 ml of milk check it out using the text convert plugin. Okay. Now the status is off. I would choose the on. You can adjust the mark here for the okay, and then you will use the pointing finger to put it on top of the tab and the other two fingers to hold the texture. Then we will put it into the middle of the mixer and let it mix for about 10 seconds. You finish? Turn it off, take it out. Then you will do the same for the next dilution, 10 to the power minus 2. Make another sterilized tip. As quickly as possible. Okay, now you take 1 ml of the 10 to the power minus 1 solution. Open the cap, drain the tube, and pipette 1 ml. Drain it twice, close the cap, and take the 10 to the power minus 2. Open the cap, drain twice, and release the drain twice, then choke. Expose the micropipette. Same. Turn it on. Another speed. Pinch it for 10 seconds. Off. Now, take another tip. Set it. Open it. Drain twice. Take up 1 ml. Careful. Close it. Take the tube, 10 to the power minus 3. Open it. Drain twice.
Okay, so this is how we take a liquid sample and we do the zero dilutions. Yeah. For solid sample, heterogeneous sample, we also make a good distribution of microorganism by mix together the sample with the diluent, like previous video Miss Ying this sampling of grapes and she mixed together grape and baking water in a blender. Yeah. But in industry, we will use another equipment to make a good distribution of a solid sample. We call stomacher. In our lab, we don't have stomacher, but I would like to show you what a stomacher is.
imagine. It's a beautiful thing. No astronaut launches for space with their fingers.
Seward, with offices in the UK and North America, are manufacturers of the one and only Stomacher laboratory blender. The Stomacher is the leading laboratory blender available in the market, globally referenced for thousands of protocols within the food industry. The Stomacher is easy to use, durable and reliable. It allows for easy and effective processing of multiple samples without the risk of cross-contamination. Complete with easy-to-use digital display, the control panel allows simple adjustment of processing time and paddle speeds, with auto-run and programmable features. The Stomacher 400 circulator and wide range of accessories is capable of processing a large variety of food sample for analysis, such as meat, seafood, vegetables, fruit, cheese, and pastry. The patented circulator paddle design provides enhanced organism recovery with reduced debris production. Seward has created a wide range of Stomacher bags and accessories to aid sample preparation, including bag clips, bag racks for storage and incubation of samples. Stomacher strainer bags contain a robust strainer element, making it easier to pipette certain samples. Stomacher closure bags are easy to use and provide an ideal low-cost alternative method for storage and incubation of samples. Stomachers are full access to the paddle chamber is gained by opening the door assembly, clean with laboratory disinfectant. With a large warehousing facility, most products are shipped directly from stock, allowing short lead times. All Stomachers are proudly built in the UK and manufactured to ISO 9001 standards. Seward's 15-point quality control checks guarantee reliability, consistency, and traceability, allowing us to offer a comprehensive 3-year warranty on Stomacher machines. Ongoing product service support is provided by our well-trained staff, combined with our global logistics partners and comprehensively stocked.
Yeah, so another equipment that we can use to homogenize.
Seward, with offices in the UK and North America, are manufacturers of the one and only Stomacher laboratory blender. The Stomacher is the
Yes, Stomacher is used to homogenize solid sample. Yeah, the dilution series I am missing already demonstrate how we can do the dilution series. Yeah, and a serial dilution is a series of sequential dilution used to reduce dense culture of cell to more usable concentration. Each dilution will reduce concentration of bacteria by a specific amount. Yeah, this is the sample. Yeah, and yeah, we can assume this is the liquid sample. Yeah? And if we want to make 10 to the power minus 1, yeah, the first dilution, we just take 1 milliliter of sample into the test tube. The test inside the test tube already contain 9 milliliters of diluent. Yeah, and we follow the 9-fold. Here, 1 cell of course we need 9 milliliters here. And with this dilution, when you incubate on Petri dish, you can see that too many colonies, you cannot count for the first dilution. Yeah. So you need to reduce further dilution so that you can count easily. Okay. And yeah, this is what I demonstrated in the video, that dilution are usually performed in series of 110 uh, 100 dilution. Yeah. 110 that consists of 1 milliliter of volume sample, yeah, added to 9 milliliter volume of diluent. Yeah, you can see here we get, if we start to take 1 milliliter of sample, so we add 9 milliliter of diluent. And if you add 11 milliliter of sample, so of course nifold here we need 99 milliliter of diluent. Yeah. Okay, there is a question here. How much diluent do you usually use to dilute your sample? This is what I want you to repeat again. No, nothing new.
Yeah. Maybe we will dilute in 10 power minus 3 or 4, maybe 3 or 4 diluent.
No, I don't ask you how many dilution series that we need. I just ask how much diluent.
Usually I use 2 diluent. 2
How much?
2.
No.
2.
How much?
I
The
think
amount.
we apply the nifold quantity.
Yes, nifold quantity. Who is answering?
My name is Huan An.
Yeah, 1 plus gram. Yeah, nifold quantity, of course. Yeah. We don't care, yeah, how much sample that we take, but we care nifold. Yeah. If 1 milliliter of sample, so called nifold, we need 9 milliliter. Yeah. If 20 gram of sample, of course, we need 225 milliliter. Okay. Can you answer again? Why do we need to do dilution?
We need to make the dilution for like dilute the sample to be less than, to be easier to count the number of the microorganism.
Yes. Quỳnh again?
Yes.
Okay, so 1 plus score for Quỳnh. Yeah, we need to do dilution because we want to reduce the amount, the concentration of microorganism. If we reduce the amount of microorganisms, so it's easier for us to count. Okay, so dilution already finished. Now we gonna to talk about another technique during microorganism analysis. There are 3 techniques related to prejudice. What are they? They are pour play, spread play, and stacking. Yeah, there are 3 techniques here. Yeah, so that's why in the first exercise, I ask you to calculate to identify the amount of diluent, of medium, we need for like BCA, BCA. We use 3 techniques for BCA, so that's why the amount of medium differently for different techniques. For pour play and spread play, talking about similarity for both techniques here, that this is the technique we use to count, colonies, to count microorganism colony on Petri dish. Yes. That is the similarity between two techniques here. How about stacking? Yeah, stacking, we don't use to count. We don't use to count colony, but we use tracking technique to isolate colony. Yeah, to isolate colony, because on a batch disk, so there are many colonies. So if you want to take that colony for further analysis, so we just isolate them. Yeah, isolate them. You can see here, you can see clearer colony here. If you want to pick up one of them, it's easier than from easier than this area. This area is too crowded, so you cannot take the build colony. Yes. Okay, so Boole plate and spread plate is used to count colony. Now you remember this similarity and the difference. You can see here on the left and on the right the difference. For Boole plate, the volume of sample, yeah, you test sample, yeah, from the tester, yeah, after you dilution, yeah, from the tester, you add on the batch disk. Yes, for Boole plate, you need 1 milliliter of sample, but for spread plate, you need less than. You need only 0.1, 0.1, and in the batch disk here, yeah, you add 1 milliliter of sample to empty batch disk. There is no medium inside. It's empty. You just add 1 milliliter of sample. Yeah. After that, you will pour the media. Yeah. It's cover the sample. Yeah. The sample inside and the media, yeah, above. Yeah. Differently in spread plate, you have the battery already containing the medium, and you add sample on the surface of the medium. Yeah, so that's why for this you need only 0.1. You don't need more. Yeah, but for Boole plate you need more, because after pouring medium, the medium will mix together with your sample, so you need a huge, big amount of sample. Yeah, it's easier for detection. Yeah, but for this one, you just use this is a spreader. You spread on the surface, so it's easy to recognize colony. But for this one, you just mix up. Yeah, so if you add low amount of sample, and maybe it cannot distribute evenly on the batch disk, so that's why you need a larger amount of sample. Yeah, the difference here, after you add medium, yeah, into the batch disk and you swirl, yeah, you swirl. For spread plate, you don't need to swirl. Yeah, you swirl the medium to mix up the medium together with sample. After incubation, you will see the colony growing and on the solidified medium. So both the colony inside and the colony on the surface. Yeah, and most of the time this technique to detect anaerobic microorganism. Yeah, for this technique spread plate, after adding sample, you use a spreader. Spreader here need to be sterilized, of course. Yeah, but not too hot. Not too hot. If it's too hot, it can kill microorganism. Yeah, and you use spreader to spread the sample over surface of the medium evenly. After incubation, you will detect colony on the surface only. on the surface only. And this technique is used to detect aerobic microorganism. Yeah. And so if I ask what is the difference between them, you can answer this. Okay. Uh, this is the demonstration video how to do the pour play technique.
Okay, so now I'll show you how to do the pour plating technique. So first, what is a backyard pants and a walking area. Right. So before me, I have a bottle of BCA cold enough to 40 to 50 Celsius degree, so that it's not too hot to kill a microorganism. And we will do pouring for dilutions 10 to the power minus 1 to 10 to the power minus 4. And a sterilized petri dish with label. And I label here 10 to the power minus 1 milk sample, BCA, as the medium, 37 Celsius degree as the incubation temperature, the group name, the date of inoculation, and the technique we have. Okay, so in total we have 3 techniques: spreading, and pouring. This one is pouring. And a box of sterilized pipette and a micropipette. So for this one, we will transfer 1 ml of the dilution into the petri dish first. Then we will pour the agar into the petri dish. Okay, so I use the 1 ml micropipette. Now I will lie on the alcohol bottle. I will take a tea aseptically. This one is the 10 to the power minus 1 dilution. I open it using my little finger and my left hand. Then I flame it twice. And then take 1 ml of the dilution. Flame twice. Now close the cap. Then I will transfer 1 ml into the middle of the petri dish. Make sure that you do not open the petri dish so widely. Just parts of it to avoid contamination. And I just close the tip, close the petri dish. Hang on the micropipette. Now move to the medium. So make sure that the medium is hot enough, warm enough, so that it's not solidified as well. It's not too hot to kill the microorganisms. So first open the cap, flame the neck of the bottle twice, and then I pour 2/3 of the petri dish. Now I close. Now I will close the dish, and then I will rotate the petri dish 5 times clockwise. 5 times anti-clockwise. Make sure that you don't rotate it too hard so that the media won't get out of the petri dish. And 5 times vertical. And 5 times horizontal. Now it's finished. You will open the lid, leave it to cool down. And when it's solidified, Then we will seal the dish with a veggie film.
Okay, so in this video, there is one mistake that we did on purpose. Can you recognize what mistake? Who can recognize? If you can recognize this, you can get 5+ score. No one? When you test sample, you can see when Miss Duyên take 1 milliliter of sample, and she didn't do something. Okay, I can go back and you can tell me. Yeah, I indicate exactly when she takes sample.
We will do pouring four dilutions, 10 to the power minus 1 to 10 to the power minus 4. And I sterilize the empty dish with label. And I label here 10 to the power minus 1, no sample, ECA as the medium, 37 Celsius degree as the incubation temperature, the group name, the date of inoculation, and the technique we have. Okay, so in total we have 3 techniques: spreading, and pouring. This one is pouring. And a box of sterilized pipette and a micro pipette. So for this one, we will transfer 1 ml of the dilution into the veggie dish first. Then we will pour the agar into the veggie dish. Okay, so I use the 1 ml microphone. Now I will lie on the alcohol bottle. I will take a tea respectively. This one is the 10 to the power minus 1 dilution. I open it. This is my little finger and my left hand. Then I flame it twice. And I take 1 ml of the dilution. Flame it twice. Close the cap. Then I will transfer 1 ml into the middle of the veggie dish. Make sure that you do not open the veggie dish so widely. This part of it to avoid contamination. And I dispose the tea. Close the veggie dish. Hang on the micro pipette. Now move to the medium. So make sure that the medium is hot enough, warm enough, so that it's not solidified as well. It's not too hot to kill the microorganisms. So first open the cap, flame the neck of the bottle twice, and then pour 2/3 of the veggie dish.
Okay, so someone recognize the mistakes?
Yes. She didn't do the vortex for the sample.
Yeah, that's right. Yeah, that's right. Yeah, we need to distribute microorganism evenly. So we need to vortex before you take 1 milliliter of sample. Is it fact?
Yes.
Okay, fact. Because you have to watch the second time, so you get 3+ score. Okay. Yeah. Yeah, so that's it for pour plate method. Now we move to spread plate method.
Okay, so now I will show you how to do the spreading technique. Now for the spreading, we will incorporate 0.1 ml of the dilution, 10 to the power minus 1 to 10 to the power minus 4. Here I will demonstrate with 10 to the power minus 1 dilution. And this is my BCI dishes, sterilized one, solidified one. And this is a spreader. It's a triangle. And a box of sterilized tip, the special one for the micro pipette, 0.2 milliliter. Okay. So because we will transfer 0.1 ml of the solution only, so I will adjust it back to 0.1, which is 100. So again, first I will disinfect my hands and the working area. I already have a alcohol burner. Yeah. I will unseal one of the dish and insert it. The dish is already labeled with the technique, the sample, and every information, all information. This is my 10 to the power minus 1 dilution. First, I will vortex it to make the solution evenly distributed. I will vortex it for about 10 seconds. Get it out. Effectively take out one tip. Now I take 0.1 ml of the dilution. And then transfer it to the middle of the agar surface. Just touch the tip. Now I will use the spreader to spread the sample evenly. But first I have to sterilize it by flaming it 5 times for each side. Now I will leave it aside, but near the flame for it to cool down. Because if it's too hot when you touch your sample, it will kill all the microorganisms inside the sample. So then I leave it for a while. Okay, now before we use it, we have to test whether it's cool enough or not. So we will touch one angle of the spreader to the edge of the agar surface. And there's no sound. If you hear some sound like zip, zip, so that is too hot. Another way is that you will touch a side of the spreader to the lid of the petri dish and use your hands to feel it color is hot and hot. Still hot, so I will wait a little bit. Okay, assume that it's cool now. Assume that it's cool now. I can check. Yeah, assume that it's cool now, and I will do the spreading. So I use the front side to spread it 5 times horizontal. And then clockwise. One hand, one finger will take the bottom of the fish, and the other finger and 5 times anti-clockwise. Yeah, this is done. Make sure that you do not over the heat too wide like spreading, because the microorganisms in the environment may contain your sample. Now when it's done, we close the lid, and then we flame the spreader again to sterilize it before we start with the next sample. 5 times for each side. Okay. Make it aside. For this one, we will seal it. Let me see. Seal around. And then we will import the vegetables. After we finish, we will incubate this in an incubator at 37 Celsius degree.
Okay, so two technique, both play and spread play. Now striking technique. So for striking technique, as I mentioned that we need this technique to isolate colony. Yeah. So we start the first area we call the this is the first number one. The first area. Yeah. We make some lines like this, and we rotate the prejudice 90 degree. And then, we start from the area, the first area, and move to the second area. Continue until the fourth area. After incubation, the fourth are we call the quarant area. After incubation, we will get colony isolation like this. Yeah, this is the demonstration.
Okay, so now I will show you how to do the striking technique. So first of all, use the front hand. For the striking technique, we will inoculate this loop using the flame. Okay, until it's red hot. It's red here. So now I move to the next section. So that I will sterilize the whole area. It's already red hot now, so I move it aside to let it cool down. But leave it near the flame. Okay. So for the loop, so you will hold the loop as you hold a pen. Yeah. Like this. So you do not hold it like this or pointing it to your body. Just hold it away from your body, but near the flame, and like holding a pen. Okay. After a while, I think it's cool now, but to make sure that it's not too hot to kill the microorganism, I will touch it to the edge of my PCA, or touch it to the leaf of the vegetables to see whether it's cool or not. Yeah, it's cool enough. Now I will open my milk bottle, flame twice, and I will take a loopful of the sample. Flame twice, close the bottle. Okay. Now we will start the stretching. So in your mind, imagine that the veggie dish will be divided into 4 quarters. 1, 2, 3, and 4. I will start with the 9 o'clock point here, and I will start stretching the zigzag line until the 12 o'clock here, and I will rotate and continue. So first, yeah, just open the lid this one, but I will, to show you clearer, I will open it wider. Start from the 9 o'clock, then you stretch it, draw it, to the 12 o'clock, then you rotate 90 Celsius degree, and from that point, you start the second track. Close it, rotate the third track, and the last one. In the middle, there may be some space, so you just make some dots. So make sure that the whole area is covered with your stretching. Yeah, do not just stretch at some area and leave some empty space in the middle or somewhere, because it will waste a lot of our medium. Okay. After we're finished, we will sterilize the loop one more time until it's red hot. Okay, it's red now. I will move to the side. Then we'll leave the lid aside. And now I will stick it with BET. And the same, we will store the BCA upside down, inverting the vegetables so that the moisture will accumulate on the lid of the vegetables. So one more thing to remind you that do not do the stretching too hard, so the agar surface will be broken and you won't get any good results.
Okay, so from this video, can you tell me why Miss Yuen use sample from the bottle? She don't use sample from the dilutions.
So because of the purpose of streaking, we want to isolate the colony, right? So
Yeah.
if we take from the diluent sample, the concentrations of our microorganism will be very less compared to the bottle. So we will streak it from the high con- high density of microorganism, and then we keep streaking until the end point, which will be the less, which will be the less
Yeah, are you
density
here?
of, yes.
Yeah, very good. He's answered correctly. Yeah, he one plus score. Yeah, we just need to isolate microorganism. We don't want to count, so we don't need to use a dilution. Yeah, use the sample from dilution. Yeah, sample from dilution can have a less dense amount of microorganism. Yeah, it's yeah we need a dense uh, amount of microorganism that easier to isolate. Okay, so we have four area of isolation. So which area will have the least amount of microorganism?
I think the area we have a list microorganism is the fourth one.
Yeah, correctly. Fact, you can get one plus score. Okay, so there are many patterns of striking. Yeah, not only quadrant. Yeah, quadrant like this quadrant. We have four area. Another, the popular one is this one, quadrant. Yeah, quadrant method. It's easier to isolate. Okay, this is just what Miss Yun already demonstrated when she do the bull play technique. Yeah, she mix sample together with medium, vertically 5 times. Yeah, she move the battery disk vertically 5 times. Yeah, and clockwise 5 times. Yeah, horizontal anti-clockwise. Yeah, to make a good mixing. Yeah, between medium and sample in bull play technique. Yeah, this is some culture transfer instrument. Yeah, we have loop, we have a needle. Yeah, like Miss Yun have just mentioned how she do the striking technique. She uses a loop. Yes, and this is the cabinet that we can use to uh, do culture. Yes, yeah. During in this cabinet, you know that it's in a sterile environment. Yeah, it can absorb some not good things like gas. Some microorganism. And this is incubator. Yeah, incubator we can control the temperature, the humidity, something like that. Okay. After incubation, we either incubate in a petri dish or incubate in a test tube. The result after incubation we can have like this. In petri dish, this is the isolate technique, striking technique. You can see colony like this. Yeah. The growth of microorganism in liquid, liquid media in the test tube, you can recognize uh, the turbidity. Yeah, it's more turbidity. That means more turbidity than this one. So that means there is a growth of microorganism. Yeah, or you can see the layer of microorganism on the surface or at the bottom. Yeah. Yeah, some signal to recognize growth of microorganism you can base on that. Okay, so in the previous slide, show you how to culture microorganism from sample, yeah, to medium. Now we have another technique is subculture. Yeah, subculture we don't culture from sample, but we culture from one medium to another medium. We call subculture. Yeah, a subculture is a new cell or microbiological culture made by transferring some or all cell from a previous culture to the fresh grow medium. Why do we need to do subculture? This action, yeah, is used to prolong the life or expand the number of cell or microorganism in the culture. Yeah, if you want to prolong the life of microorganism or you want to expand number of microorganism. Yeah. So there are three main subculture technique. Use subculture from liquid medium to solid liquid medium. Use subculture from a solid to liquid. Use subculture from solid to solid. Yeah, I can show you how people do a subculture technique.
One of the things we sometimes need to do is to take a culture of microorganisms and sub it into a number of sterile broths. So we've got tubes for the different groups in the class. And so what we need to do is to take a loopful of that culture and inoculate each one of those. Now, I believe you're quite good at that. So I was going to get you to demonstrate how you would do it.
Okay, well, the method that I use, some might say is a bit cheating, but I find that I have quite small hands, so I'm going to show yours, we'll show this a bit more different. So I'm going to start off just by putting a bit small to the blue frame.
Yes.
Use.
Right.
Nhi quậy lấy giùm coi.
Right.
Rồi.
Like that, cool. Now, I'm going to just give the tube a little shake, just stir up the culture a little bit.
Why is that?
Well, Bacillus subtilis, it clumps into what they call it a pellicles, like
Right.
grows. But some teachers sometimes find that because it clumps, it's maybe not growing properly, but that's just the way Bacillus subtilis grows.
Grows, yes. Yeah.
So by just disrupting it, we'll get some of the cells just to fly off into solution, which is what we want.
Right.
So that I can now flame the lid again, the mouth and the tube. Just take a loop full out, same as before. On, and now I want to take my sterile broth,
Yeah.
get the lid off this one,
Yes.
and then in my loop full of culture, just give it a little shake around there. Flame it again. Like that. It's a little bit of a cumbersome way of doing it. It takes a little bit longer than I'm sure the method that you're about to show us, but I find it's a bit easier and more reliable for myself.
Okay. Well, I've got a method where I try and hold both tubes in one hand and save myself a few seconds. Right. So let's have a look. I've got the culture here, yes, and I've got the broth there. And what I do is I lay them along my fingers. You can actually hold them with your fingers, but it's a little bit reassuring just to put your thumb there. And I've got a bit of a gap between the two. I take the inoculating loop and I sterilise the loop so that it's nice and red. and I go for the far cap. I take it off, come back for that one, flame them, go into the culture like that, and then simply go into the sterile broth. I'll give it a little bit of shake, but nothing too busy. And then what I do is flame the two, put the cap back on, that one back on there like that, sterilize the loop so that it gets nice and red, and I've saved myself all of a couple of seconds.
Excellent.
And I can do the next 20 for the class.
Okay. In this video, who can show me that there is some error? There is some error that we need to avoid.
As I can see, it's mostly about the accepted technique. So
Yes.
So they don't wear the glove, for example, and they don't sterilize the environment when they work. And
Yes.
also the very important part, which is the mask. We always need to wear the mask when we work with the microbiology.
Yeah, very good. Are you Huy?
Yes.
Okay, Huy, 1+ score. Yeah, they don't wear clothes, they don't wear mask. Yeah, they don't sterilize the working area. Yeah, and even the look after using, they put down on the table. It's not good. It's not good. Yeah. Okay. That's it. The subculture from liquid to liquid. Yeah. Subculture from liquid to solid or liquid. Yeah, like the video show. First, you immerse the sterile loop in the fluid to be subculture. Yeah, here. Yeah, remember, don't touch the wall of the tester. Yeah, when you take out from the first medium, yeah, either inoculate a loopful of fluid on a appropriate agar plate, or stretching out for individual colonies, or inoculate with a loopful to further fluid culture media. Here, yeah, from liquid to liquid. Yeah, we put in the new medium, and try to avoid contact. Yeah, to contact on the wall. Yeah, before culture of the broth that may contain microorganisms, including anaerobe, gently agitate. Yeah, you can see in the video the woman. Yeah, she agitate the first step a little bit. Yeah, to give an even distribution of organism throughout the broth, with as little disturbance as possible. We don't use a vortex here. Why don't we use a vortex here? Who can answer? Yeah, you can see in the video the woman, she only agitate. She don't use a vortex.
I think it may relate to the distribution of the microorganism in the test tube. So I think that for the microorganism, they usually not distribute evenly on the test tube. So if we detect which part of the test tube contain it, so we only inject our needle to that area, to that space, to get the microorganism. I think so.
No, you can see this with as little disturbance as possible. If we use the vortex, so we have a big disturbance, because here already there is living microorganism. Yeah, it's not like in the sample. Yeah, there is some living, yeah, living one. Yeah, microorganism. So if you use a very strong, yeah, force, yeah, I will use a very big disturbance. Microorganism can be influenced. Yeah. So as I told the other student, when you're working with microorganism, like you are loving a girl, yeah, like a boy loving a girl, you should take care of them. You take care of a girl. A girl is like a rose, yeah. You always, yeah, do the gently, yeah. You don't want to make them um to be killed. Yes, like a girl, yeah, very sensitive, yeah. So you love them very much, love the girl very much. Uh, so try to avoid uh, any hurt hint to her, for example. So here, yeah, because you don't do experiment directly, so I can say that. But some students last time uh, they did very strong action, so they don't detect any microorganism. Yeah, there was no microorganism because they did very strongly. Yeah, very strongly. So be careful when you already have microorganism. Don't kill them. Yeah, don't hurt them. Yeah. Okay, so this is the subculture from liquid to liquid. How about subculture from solid? From solid. This is the solid medium. Yeah, battery this. You have already some isolated microorganism. There are two microorganisms, the red one and the blue one. Yeah, already isolated. If you want to take, you want to subculture the red colony here, you just use the sterilized loop and pick up the red colony here, yeah, and then you transfer to a battery, the new medium, yeah. You also do the tracking in the new medium, or you transfer this loop into the liquid, yeah, liquid medium, like you do for the first method here, yeah. And yeah, if you want to subculture the blue one, just take the blue one, yeah. Okay, that's the subculture technique. Another technique that we should take care of is a stopping technique. Yeah. So in microorganism analysis, besides you count colony, yeah, you can also detect presence of microorganism. Yeah. Let me see. I can check if I have some information in the is here. Okay. So if there is criteria related to microorganism for one product, this is what products here? This is uh, the suffering, yeah, wake up cola, yeah, suffering. Uh, there is some criteria for this product related to microorganism. I talking about microorganism criteria only. Yeah, you can see the total aerobic count, the limit Here it's 10 to the power 2 CFU per milliliter. Yeah. E. coli, there is no value here. It's not present. Yeah. For this one, the total aerobic count, you can use counting technique. Yeah, you can use like BCA. Yeah, BCA to incubate microorganism from sample, and then you count. You do some dilution, incubation, and then you count. You count if the number of colony is less than this, it's okay. Yeah, or coliform the same. But for E. coli, it is asked to be not present in the product, so you cannot count. If you count, there is no colony, zero colony, you cannot conclude that there is no colony here. Yeah, if you use counting technique indicate you the number of colony, but it not indicate you that there is no colonies. Yeah, there is no microorganism in your product. Yeah, zero doesn't mean, when you count it's zero, but this is zero, it doesn't mean that there is no that kind of microorganism. So you cannot use a counting technique. You use another technique to detect if E. coli present or not in your product. So with this technique, we use some bio reaction. So if there is those microorganisms in your sample, this microorganism tends to the nutrients, tends to some compound in the medium, they will react. Yeah, there is some reaction. They will react, and based on those reaction, based on those signal, maybe the red color, the black color, or something like that. So you say, okay, there is that kind of microorganism present in our sample. Yeah. So this is we call biochemical test. Yeah. Biochemical test, we don't use a battery disk. Yeah. We use a test tube, and it use solid medium. Remember, solid medium here. And we have to subculture from one medium to the new medium here. And based on this new medium and some signal, you can say, okay, this sample contain, for example, E. coli. Contain E. coli. There is E. coli in that sample. So we can, because there is E. coli, so we can reject that sample. It's not allowed to be in the supermarket. Yeah, it's not allowed for consuming. Yeah. Okay, so for biochemical test, we use the stopping technique. Yeah, stopping technique. For this technique, we have the medium, but it should be in slang. We call slang culture. You can see it's slang here. The surface is like a slope like this. Yeah, slope like this. So to make it to make slang culture like this, after you pour the medium, the agar inside the test tube, and you put down the test tube, it's not completely down. Yeah, it's around 45 degrees down, and then you will have a slang like this. Yeah, it's solid. Remember. Yeah, after solidify, you will have a slang culture like this, and then you do the stopping. Yeah, first you sterilize. For stopping, you don't use a loop. You use a needle. You can see it's needle here. You sterilize the needle and pick up one well isolated colony. Yeah, you have the battery disk. with isolated colony. Yeah, that's stacking technique help you to isolate, and you pick one colony from Petris, and then you stop inside this agar media. Yeah, stop until here, not to the bottom, not to the bottom. It's around 2 centimeters from the bottom. Yeah, you stop the needle inside the agar medium until here. Yeah, and you stop. You don't go further, and you lift out the needle from the medium. On the surface, you will make zigzag. Yeah, you also do the stretching technique on the surface. You make the zigzag line on the surface, and then yeah, you after that you close the cap but loosely and incubate this medium. After incubation, you will see some signal from this medium, and you can say, okay, that kind of microorganism present in your sample. Yeah, this is the demonstration how we do the stopping.
Okay, so now I will show you how to do the stuffing technique. So for stuffing, we will have two samples. One is a chicken sample. The nutrient agar with chicken sample, microorganisms from the chicken. And another one is the nutrient agar interpolated with the salmonella strain. So here I have a tip of TSI plant. In needle, this one is the needle. It's different from the loop because of the pointed. Okay, so first of all, disinfect my hands. So from this lab, you will have 5 tubes of TSI. 3 of them will be inoculated using a stuffing technique with the chicken sample. 2 of them you will inoculate it from the salmonella sample. So first I will lie up the alcohol burner. Unseal my So when you working with salmonella, pay extra attention because this one is not a beneficial microorganism. It's a kind of pathogen. So if you careless, you may contaminate yourself with this kind of microorganism. So again, first I will need to sterilize this needle until it's red hot, the whole bed. It's red hot now, so I move to the next section. Okay, it's already, so I leave it aside for it to cool down. So like the loop, you will hold the needle like you hold in a pen. Yeah, do not hold it like this or pointing it directly to your finger. Pointing it out, but also gently. Okay, leave it aside a while for it to cool down. So to check the temperature of the needle, so use the leaf, touch the needle to the leaf of your petri dish. It's already cooled down, so you now can pick up a colony, a typical colony on the surface of the agar. So now, for example, you pick up a small amount of the colony. Just swab it on the surface. Do not point it through the agar and destroy the agar. Okay, just point it on the surface. small amount. Then you take it out, do not sterilize. Now take out your TSI tube, flame the neck of the tube, and now we will do the stamping. So first I will point the needle straight to the bottom of the tube, but not to this bottom, just around here. Yeah, right in the middle. And then I will do the striking on the flat surface. Okay. Make sure that the needle don't touch the wall of your test tube. Now I point it to the surface. Okay, I will stop here, and then I will remove the needle the right way, the right route. I point it inside. Do not point one side and then take it out another side, so that it will break the TSI gap. Now, on the surface, I will do some stretching from the bottom here to the top of the slat. When I finish, I take out the needle, flame the neck of the tube, close the cap, and sterilize it again. Make sure that the whole needle is sterilized until it's red hot, so that the microorganism will not survive. After the sign, now I will seal this dish with the vegan cream. For this tip, I will collect all the tip into one rack, and then we will tightly seal the rack. Put it inside a nylon bag and incubate the sample.
Okay, so that is the stamping technique. Yeah. The final part today we will learn is a plate count method. How can you count colony? Yeah, and we use a unit to for the result of your counting technique. We call colony forming unit, CFU. Yeah, as I show you in the microorganism analysis, microorganism criteria of sovereign. Here you can see the units here, CFU per milliliter. If your sample is liquid, this is sovereign. Yes, so you use CFU per milliliter here. Okay, so colony forming unit is a unit used to estimate number of viable. Yeah, viable mean living. Yeah, it's not that viable bacterial cells in a sample. And the number of CFU here equal to number of bacteria over milliliter or gram. Yeah. The standard plate count method consists of diluting a sample with sterile saline, sterile diluent, until the bacteria are dilute enough to count accurately. This is a formula that you can calculate. You have a different battery days from different dilution. You will take the average, take the average number of colony from duplicate place. Any analysis you always repeat. You always do the replication. You never make one sample for one factor. So that's why when you do the replication, you will take average for any analysis. Yeah, in food analysis, you also take do the repetition, so you will take the average. Yeah. So here is the same. You take the average from duplicate, triplicate also average. Yeah, the average number, and divide by dilution factor. Yeah, for which dilution you get the dilution factor, and the volume plated. Remember, for Bohr plate technique, you use 1 milliliter. So you, if Bohr plate technique, you multiply by 1 ml. Yeah. If for spread plate technique, you will multiply by 0.1. Yeah, 0.1. This is the formula to calculate the number of colonies. Yeah, I wanted to show the video how you count colonies.
So after we incubate our sample, our baby dishes, then we will move on to the counting step. So now I will show you how to use this one, a colony counter. So here we have a PCA dish, which is already incubated. So first, I will look in, and then I will switch on. So the light already turned on, and this is a microscope for you to observe even the tiny colony. And this one is the panel where you place your battery dish on with the red lights already here. And this side we have the number, which is your number of colonies. Okay. For the PCA dish, first I will unseat the dish so that I will see the colonies clearer. Because some of the colonies grow at the edge of the petri dishes, so that if we move, if we remove the film, we will see it clearer. So I also remove the label here using alcohol. I will spray some alcohol onto the tissue and swipe out the label. But make sure that you remember the dilution factor, the technique, your information. Okay. So to see it in the most clearest way, we will invert the dish. Not this one, because the moisture will condense to the lid. So we will invert the dish and leave it on the counting panel. Okay. You also do not open the lid, because the microorganism inside may contaminate your environment and maybe your body. Okay, do not open the petri dishes. Okay, so you leave the dish here. For each colony, you will use your marker, look through the microscope and point onto the colony to signal that you count that colony already. The number will appear here for you so that you do not count yourself. Okay, we just if you miss any points, you can use these two buttons to add up or subtract the number. Okay. So when you finish with one dish, flip it aside, then you press reset. So it's back to 0 and you can start with the next dish. The finished one, you will label it again and seal with the the B effect. Yes.
Okay, so that is the method how you count colony and to spread your result. When you count, you have, for example, 16,545 CFU per milliliter. You won't spread the result like this. No, never. Yeah. First, this is 5. Yeah, 500 something. So it's more than 5. So you can round up this number first. You round up. So it become 17,000. Yeah, 17,000. And you want spread 17,000 CFU per milliliter. You can see in the microorganism criteria, they always spread like this. Yeah, they don't spread 100. They spread 10 to the power 2. Yeah, the limit colony. Yeah, don't spread 100 like this. Yeah, 100 just 10 to the power 2. So if after you round off this number, yeah, it will be 17,000. Yeah, and you always spread the result and the number between 1 to 9.9. Yeah, so 17. So you can spread is 1.7. 1.7 multiply 10 to the power minus 4. Yeah, it should be like this. Yeah, so the first figure, you use the number between 1 to 9.9, not more than 9.9. Yeah, you cannot say 17 here and multiply 10 to the power 3. No, you should spread like this. Okay, this is the way that you spread your result. And there are some rules that you can follow when you count the colony. Okay, so please, all of you, can you open your lab manual and open the page related to counting rule. I will pick up some person who will read louder, please, the rule for me. Please open your lab manual first. Okay, Chu Bảo Khuê, can you read rule 1 for me?
Yes, teacher.
Yes.
So the rule 1, colony in the range of 20 to 200. So if only one plate with a colony count in the range of 20 to 200, calculate the number of colonies in that plate instead of an average in equation 3.1. Yeah, so if plate from 2 consecutive dilution yield from 20 to 200 colony, compute the number of colony for each of the 2 dilution. If they are not aggressively different, average the number. If they are substantially different, report only the lower computed.
Okay, yeah, thank you, Khuê. Yes, so look at the rule 1. Rule 1. Rule 1. If you see the colony in the range between 20 and 200, how can you spread your result? So if only one plate with colony count in the range, so you calculate the number of colony in that plate. Yeah. So if you do the replication, yeah, you repeat for one dilution, you have two prejudice, but only one prejudice have the colony. The other one, there is no colony. So you just check the number of colony in that prejudice only. Yeah. So you don't need to take average. Yeah. If you take for average, for example, the first play is 21 colony, the second play is 0. If you take average between 21 and 0, it's not good. Understand here? Okay, so you just take 21 only. For 0, you don't count. You don't need them. Yeah, it's 0, so don't take the average. But both prejudice have the number. For example, the first one is 21, the second one is 30. So you will take the average between 21 and 30. Okay? The second thing in rule 1, that if play from two consecutive dilution, you 20 and 200, you will compute the number of colony for each two dilution. Yeah. Let me see. This is an example. So, the first example is not in rule 1. You can see 0, 0 on of dilution 0. So you don't apply rule 1 in this example. Yeah. Example 2 is less than 20, because you remember that rule 1 in the range between 20 and 200. So for example 2, is the number of colony is less than 20, you don't apply rule 1 in this example. Yeah. For this example, one dilution is more than 200, but the other dilution is in the range between 20 and 200. So we can apply rule 1 here. Yeah, we can apply rule 1 here. This is out of the range. We don't use it. 3 is less than 20. It's also out of the range. We don't use it. So we only take this one, 45. It is between the range of 20 and 200. So you take 45. Divide by. So remember the formula. This is the dilution factor. Look at this. This is 10 to the power minus 6. So you write 10 to the power minus 6 here. Multiply the volume. You can see this is they take 0.1 from the test tube on the petri dish. Yeah, this is the spreading technique. So we multiply by 0.1. And you calculate, of course, you have to spread like this, 4.5 multiply 10 to the power 8 CFU milliliter. Okay? Yeah. So, this is we can apply rule 1. How about this example? This dilution contains 197 colony. It is also in the range between 20 and 200. We take it. 23 also in the range. We take it. Yeah. In the rule, look at again the rule 1. When there are colonies from two consecutive dilution. Yeah, here you can see consecutive. Here minus 5. Here minus 6. Yeah, they are close each other. Yeah, the next one. Yeah, so we can take both. Yeah, and what the rule mentioned. Yeah, you compute the number colony for each of two dilution. Yeah, so you will calculate the number colony in minus 5 first, and a colony in minus 6 second. Yeah. If they are not different, yeah, they are not so different, so big different, so you can take the average. But they are a big different. Yeah, the higher maybe three times higher than the lower one. Yeah, so you report only the lower. Yeah, so for this one, all of you, can you calculate the number of colony in dilution minus 5 and dilution minus 6? I will leave you 5 minutes to calculate, and then you can tell me what exactly the number of colony in each dilutions. Who has a final result? You can tell me. And after calculating each dilution, if you can answer, are they similar or are they big different? And then you can get 10 plus score for this question.
For the first blade, I come up with the 2 multiplied by 10 power 8.
Do you write down, or you have any calculation to show?
Oh, I'm sorry, I write on my note paper out there.
The paper, okay. You can, or can you take a photo or something like that?
Oh, so I can share you the screen and I will calculate it on the Google Docs. Is that okay?
Yes, yeah. Have you said it?
Just wait a minute to open my tab. Can you see my screen?
Yeah.
So for the first blade,
10 to the power minus 5.
Yes, of course, and we have the number, as I remember, is around 197, right? So we divide it by 10 power minus 5, which means we will multiply it for 10 power 5. And we multiply by 0.1. So
Yes.
I will round up the result. So 197 to 200, right? And we will have the result like to multiply by 10 power 8.
Okay.
In the second one, we do with the same equations, but a little bit different in the colony count with 10 power minus 6, the same fraction. And actually, it is near with the above number, also multiplied by 10 power to 8.
Yeah. So based on the rule 1, what will you do?
We will take the average because they are approximately equal to each other, not so different.
Yeah, very good, Hui. So he can get 10 plus score for his answer.
I think it will be equal to around 2.50, right?
Yeah.
By 10 power to 8.
Yes.
Yes, and that's the result.
Yeah. Good. Thank you, Hui. So, if you calculate, they are not the same, so you just take the lowest one. Yeah. Yeah. So if you don't calculate for each dilution, if you see here 197, here 23, and you can see it seems that this one is a higher double than this one. It seems that, but when you calculate using the formula, it's not double. So that's why they are similar. They are similar, and then you can take the average here. The average here. Yeah. So be careful for this one. Never just look at the number only. You have to calculate using the formula, and then you will have the final result like this. So that's it. Rule 1. Now move to root 2. Root 2. Root 2, the range is between 1 and 20 colonies. Yeah. You also apply the formula to calculate, but calculate for the lowest dilution. Yeah, the lowest dilution, and report the number as estimated. ESD. You can see the example. So, example 1. No, it's not root 2. Example 2. Yes, it's between 1 and 20. So you can take this. Yeah, can take it 12. And using the formula, it's 10 to the power minus 5 multiply 0.1. And you calculate the final result like this. And in the result, you have to show estimated ESD here for root 2. For root 2. How about root 3? Root 3, play with no colony. You calculate the result with one colony and report as less than the value of 10. Yeah, with no colony, that doesn't mean that there is no that kind of microorganism as I mentioned earlier. So you need to calculate. So example 1, you can apply root 3. 0 here, 0 here, 0 here. and you use the formula, but for the number of colony, you don't use 0, you use 1 here. And yeah, less than here. And you use the lowest dilution, minus 5 here to calculate. Multiply 0.1. And the final result is less than 1 multiply 10 to the power 6 CFU milliliter. Yeah. For rule 3, you must have this signal less than. Yeah. And you use 1 instead of 0. That's it. Rule 3. How about rule 4? Rule 4 that your colony is greater than 200. Greater than 200. A lot. There are too many colonies. Yeah, it's hard for you to count. So there is some method for you to count quickly. Yeah. We can call, there are 3 rule for A, rule for B, and rule for C. For rule for A, you will draw 1 square. Yeah. Any area on the battery disk. The battery disk, normally there are 65 square. There are 65 square like this. If you use the counter, yeah, magnifier like Miss Yuen have just showed you, you can see the square. So there are 65 square. And you choose any square with the colony and you draw 1 square. And you count the colony number inside that square. You count it. If the number of colony in that square is less than 10 colony, then you will draw 7 square horizontal like this. 1, 2, 3, 4, 5, 6, 7. And 6 square vertical like this. A, B, C, D, E, F. And you calculate colony number for each square in the 13 square here. And then you sum up the colony number in 13 square. And you will calculate like this. The sum of colony number in 13 square multiply 5 and divide by dilution factor multiply volume plated. Yeah. Can you tell me why there is 5 here? Why sum of number of square number of colony here in 13 square, but we have to multiply by 5 here.
I think because as you mentioned above, each plate they have 65 square, and so that and 13 multiplied by 13, which multiplied by 5 is 65. So we have to multiply by 5 because we just count on 13 square and we multiply by 5 to get the total of the whole dishes.
Yeah, thank you, Fat. Yeah, you get 1 plus score. Is it correctly? Yeah, we have the number of 13 square, so we multiply by 5. That means we have the total colony in 65 square. We need to know on the battery, the number of colony in the battery. We don't need only 13 square. We need everything in the battery. So we have this formula. Yeah. Rule 4B. Rule 4B. You also choose representative square. Yeah. And you draw a square. And then you calculate the number of colony in that square. If the number of that square is more than 10, yeah, more than 10, we don't apply rule 4A. We will apply another rule 4. Yeah, it's more than 10, but less than 100. So you can apply rule 4B. Yeah, rule 4B. Together with this square, you will choose three other representative square. I tell you that representative, that means the area there is colony, remember, yeah. Three other representative squares, so total you have square, four square. Uh, four square contain number of colony between 10 and 100. After counting four square, you sum up, yeah, and you divide by 4. Yeah, you divide by 4. That means you have the average for one square. Yeah, the number of colonies in one square. And then you multiply by 65 for the whole Petri dish. Yeah, the number of colonies for the whole Petri dish. And yeah, follow the formula. It's normal. We need dilution factor. We need volume. Yeah, and we have the final result. Remember, always you write the estimated here. Estimated. Yeah. And the final rule is rule for C. Rule for C. When you calculate the representative square, the number of colony here is more than 100 colony. Yeah, you don't need to count anymore. It's too much. You don't need to spend more time to count. You just write, okay, the final result is more than 6.5 multiplied 10 to the power 3 and divide dilution factor and volume related. Yeah, those are the rule that you can use to calculate to count the colonies. Yeah, remember, if you have the many Petri dish and you shouldn't start to count from the first dilution until the next dilution. You just select one base and you can count if the number of colony in this range between 20 and 200. So you don't need to count the bigger Petri dish. I mean the Petri dish with a huge amount of colony here. Yes, you don't need to count because it takes time. You don't need it because you don't use it. The priority, you just use the battery contain colony in this range. Yeah, yeah, like this example. So I have 3 dilution, and I found that this dilution have the colony number in the range 20 and 200. So I don't need to count this dilution. Yeah, I just assume, okay, it's more than 200. I don't need to count it. So when you have the final result, you don't take this value in your formula, so you don't need to count. Okay, so that is the end for today. So for the laboratory 2, you can write the report. What I mentioned in my presentation, yeah, some techniques, something like that, and I will email to you the exercise for laboratory 2, yeah, related to the plate count method, and you have to calculate. I can give you yes, like this. I will send you this file. For example, exercise 1, we have information like this, and we have the result like this, and I need you to interpret. Yeah, this is the dilution -1, -2 until -5, and you have to interpret this result. Yeah, and exercise 2, the same. You have to interpret it. Yeah, this is not countable. Yeah, too many. Yeah. Also exercise 3. Yeah. So the report for laboratory 2, you will mention all the technique, all the method that I presented for you today, including 3 exercise here for laboratory 2. Yeah. And laboratory 1, you don't need to do the report. Okay. Are you clear until now? Do you have any question?
I want to ask about the deadline for the report.
The deadline, you will submit, you have your lab work until next Saturday is the final online lab work. So you can submit 1 week later. That means December 11.
Yes. So can I ask about the outline of the report? The outline of the report, because I found it is quite a special report because we do have any number for this report. So can you give us some outlines to
Okay. Yeah.
follow for that?
Yeah, sure. So the report for laboratory 2, yeah, I will tell you for each laboratory. For laboratory 2, you have the 2 parts. One part is the technique, yeah, that I present today, yeah. You will use lab manual and use my present reference, yeah, to tell the report. Okay. The second part that is the exercise. So only 2 parts. The first part is the procedure, yeah, procedure related to techniques. The second part is the exercise. Yeah.
Okay, so for this one, do you have to do any introduction about anything like?
Yeah, if you include introduction, is very good because you will may find some reference, yeah, from internet. I need you to do more reading, more, yeah, studying, yeah. You need to discover more, not just copy from lab manual or from my presentation to your report. Yeah, so yeah,
But I can
introduction, procedure, and the exercise.
Hello,
Yes.
Miss. Can I confirm your requirement is like the literature review?
Yes.
Cover all the knowledge about the things that we have learned today.
Yes.
Oh, okay. Thank you.
Oh, that's mean the first part is literature review. Yeah, some introduction. The second part is the procedure. Yeah, how you do that technique. Yeah, and I don't need a copy one. Yeah, I need you to summarize for procedure. Summarize from Lamino, summarize from my presentation. Yeah, it should be different from what I wrote in my lab manual. Yeah, and yeah, you can find any good method to show the procedure. Yeah, and the final part is exercise. Yeah, exercise, of course, you analyze the result. Yeah, interpret the result. Any more questions?
So we need to mention about the experiment in the lab manual.
No, not experiment. the technique like I mentioned, like a bull play technique, you will describe what is the bull play technique, spread play technique, striking technique, for example. You describe, but not like the way I mentioned in my lab manual.
Yes, I got it. Thank you.
Okay. More questions? Yeah, and hopefully, yeah, for the final lab work, yeah, try to read lab manual. If you have a good answer, maybe you don't need to, yeah, to make a report. Because yeah, if you answer and you get a 100 score already, so you don't need to make a report. So try to get more plus score during my presentation. Okay. No more
Okay,
questions?
teacher.
Yes.
No, I think it's all clear.
Okay. Yeah. So thank you for your attendance. Yeah. See you next week. Bye bye.
Thank you, teacher. See you later.
Yeah, thank you, teacher. See you next week.
Thank you, teacher. See you next week.